안녕하세요. 필코리아 테크놀로지 조은진 부장입니다. 오늘 웨비나는 에디코 바이오에서 글로벌 전략 및 제품 개발을 이끌고 있는 두 분 전문가, 디렉터 오브 프로덕트 매니저인 트라비스 핫캐슬과 세일즈 앤 디스트리뷰션 매니저인 니라치 샤를 모시고 진행합니다. AI가 발달될수록 지놈 엔지니어링도 더욱 관심도 높아지고 있는데요. 그래서 시기적절하게 오늘의 주제 From Knockout to Insight Empowering Researchers with XDel, KO and Next-Gen KI Engineering으로 발표해 주시겠습니다. Hello, Travis and Nirachi. We'd love to hear your presentation.
Welcome. It's a pleasure to be here today, and thank you to the Phil Career team for giving us the opportunity to discuss CRISPR editing. So in this talk, I will start by giving an introduction to EditCo. We will explore the CRISPR workflow and technologies, and we will discuss generating knockouts and knock-ins and arrayed CRISPR screening libraries. The aim of this talk is really to give you a good introduction and grounding into generating cell models using CRISPR. But if you would like to discuss any details about your project specifically, please reach out to Phil Korea and ourselves, and we're more than happy to give you a personalized CRISPR consultation based on your project and any CRISPR issues that your lab is facing. And with that said, let's begin the presentation. So, EditCo is a CRISPR company. While EditCo Bio is a new name to the space, we have actually been around for the better part of 6 years under the brand Synthego. Synthego aimed to make CRISPR accessible to all researchers, first by commercializing the RMP method of CRISPR, which used an RNA oligo rather than a plasmid or a lentivirus. And then in 2018, it spun out, it created its own cell engineering division to allow research to perform CRISPR for researchers using their automated platform to maintain consistency. EditCo Bio is a recent spin-out of the genome engineering branch of Synthego, and we've been making custom cell models for researchers for the past 7 years around the world. We have a new brand and new investment, but EditCo Bio is still committed to really making CRISPR as accessible as possible to the research community as a whole. So between Synthego and EditCo Bio, we have been responsible for over 506,000 CRISPR edits. We get an average editing efficiency of around 80%. We've been cited in over 700 publications. And to date, we have worked with over 1,400 different cell types, from cell line iPSCs and even certain primary cells. We also collaborate with some of the top research groups and biopharma companies globally. So what does EditCo actually sell? Well, we aim to make CRISPR as accessible as possible, and we do this in two main ways. Either we can empower your lab via our CRISPR reagents, and we have reagents to help you generate knockouts and array screening libraries as easily as possible. And that's great if you want to perform the knockout yourself. Or if you want us to do the CRISPR for you, we can generate knockouts and knock-ins in immortalized cells and iPSCs, and we can perform knockout in certain primary lines. So let's dig a little deeper and talk about the CRISPR technology and the actual CRISPR workflow, which is essential for any CRISPR project. So how does CRISPR actually work? CRISPR is a kind of genome editing, which basically gives research the ability to modify DNA in any living cells. And there's been a long evolution of gene editing components, which I won't go into detail about. But the most common genome engineering technique today is CRISPR-Cas. And CRISPR-Cas9 specifically is really the most common common technique that researchers use. So how does CRISPR actually work? Well, CRISPR works by creating a double-stranded break um, and is made up of three components. First, you have the Cas9, and the Cas9 is a nuclease that cuts double-stranded DNA. You can think of it as a molecular scissors. You have the guide RNA, which acts as a molecular GPS coordinates and takes Cas9 to the right location in the genome. And then finally, you have the PAM site. The PAM site acts as a molecular docking site for the Cas9 to bind to. So the guide RNA will complex with Cas9, and this will guide it to a specific pre-programmed location and will bind to a PAM site where it will create a double-stranded break. This can result in two possible scenarios. First, you have non-homologous end joining, which usually results in an insertion or deletion mutation, also known as an indel. And this indel will most likely result in a protein knockout. Alternatively, if a repair template is present, the cells can undergo homology-directed repair, or HDR, and this allows for the insertion of a custom sequence. So, to summarize, the guide RNA and Cas9 will create a double-stranded break that results in either a protein knockout or, in the presence of a repair template, a knock-in that allows for the insertion of a custom sequence, such as a patient mutation, or a tag such as GFP or HiBiT. So there's been a long evolution of CRISPR reagents over the years. In the early days, plasmids and lentiviruses were the most common ways to perform CRISPR. Plasmids are great because they're relatively easy to use, but they typically show low editing efficiency, so not great in anything other than cell lines. Lentiviruses show good efficiency in cell lines, stem cells, and primary cells. But aside from being your lab's health and safety officer's worst nightmare, they can also integrate into the genome, and they are constitutionally expressed, which means that they can show higher off targets compared to other CRISPR methods. Um, so over time, and they can integrate into the genome, which again isn't necessarily ideal. So over time, a new CRISPR method was developed called RNPs. And here, the guide RNA is an RNA oligo, and Cas9 is introduced in a protein format. These are combined together in vitro and transfected into the cells. And the reason why this is ideal is because this method shows high editing efficiency. It's amenable to difficult to transfect cells, such as primary cells and iPSCs. And because it's a protein complex, it degrades in a few days. And so it shows far lower off-target effects compared to plasmids or lentiviruses. And as I mentioned before, all of this can be done in cell lines, iPSCs, and primary cells. So no matter what CRISPR experiment you're doing, RNPs is a viable solution. Thanks, Travis. So that is CRISPR in a whole. But let's now kind of move on to the CRISPR workflow, i.e., what your lab will be doing every time they want to perform a CRISPR experiment. And this is the workflow that we typically use here at EditCo Bio. So the first thing you have to do is design a strategy. So what are your project aims? Are you trying to generate a knockout or a knock-in? And then you can start designing the guide RNA sequences. There are lots of tools to help for this, such as ChopChop or BenchLink, and they will kind of work with you to design the guides for you. These guides can also be and then the next stage is to synthesize the guides. This can either be done in your lab, or companies like ours or Syntago can make the guides for you. The guide RNA and Cas9 is then transfected into your cell population. And this gives us what we call a CRISPR pool. So CRISPR pool is a mixed population of cells with slightly different edits, including wild type, knockout, and potentially knock-in populations if an HDL template is used. So editing efficiency is always something to consider, and it's not uncommon with difficult cell lines or knock-ins to get low editing efficiency. So we would thoroughly recommend optimizing your transfection protocols. If you're using an RMP like us, we find nucleosection to be the most effective method of delivering the RMPs into cells. At EditCo, when we work with any new cell lines, we always try up to 200 different optimization conditions to get the best editing efficiency possible for that cell line. And we would recommend researchers to try multiple optimization conditions because it's really important to get the best editing efficiency, and trust me, it will make your life a lot easier down the road when you're single cell cloning. Right, so you have transfected the guide RNA Cas9 into your cell population, and you have got a CRISPR pool. Now what? Well, historically, single cell cloning was needed for every edit, as editing efficiencies were low. But in this day and age, you're able to get highly efficient knockout and knock-in populations. And in those cases, do you really need a single cell clone? And just to kind of give you a definition, we define single cell clones by a population where 100% of that population is derived from a single cell clone, which basically means that you have an isogenic population. So going back to the question, do you need a single cell clone? The answer depends on your downstream assay. If you're looking to target genes by Western blot or qPCR, then maybe a highly enriched knocked out population would be okay. But for more sensitive approaches like RNA-seq or like proteomics, or even disease modeling, you would likely need a single cell clone. So how do you single cell clone? Well, the most common way to do this would be to perform a serial dilution and plate your cells into a 96-spot plate with an aim to have 0.5 or 1 cell per well. You will then need to look down a microscope and determine which cells have been derived from a single cell clone. These cells should be marked and then monitored as they grow out. Once grown, they should be passaged into a new plate and then QC'd genomically by sequencing. So as you can see, the single cell cloning is a long time-consuming process. And if done wrong, you can risk the wild-type cells returning and overtaking the CRISPR mutants. So that is the CRISPR workflow in a nutshell. And I would say there are two main bottlenecks to the CRISPR workflow. First is editing efficiency, and then second is the single cell cloning. And it's important to note that the higher the editing efficiency, the fewer clones you have to screen. So 90% editing efficiency will mean that theoretically 9 out of 10 clones should contain a CRISPR edit. So how does EditCo perform single cell cloning? Well, we are able to use automation. So we take the pool and use automation to plate the cells into multiple plates. And the cells are then pictured regularly, and the genotype is automated. And the final clones are validated by NGS. Next slide, please. So here you can see us taking pictures of cells from day 0 onwards to show the cells have truly been derived from a single cell clone. And as I mentioned before, one of problems going wrong with a single cell cloning is an issue that we frequently hear from customers, and it's important to get this right. Otherwise, you risk the wild type cells overtaking the mutant populations. So I've introduced CRISPR and briefly discussed the CRISPR workflow. I'm now going to go on to discussing knockouts using our XDel technology, which is currently the easiest way to generate knockouts on the market. Next slide. So just a quick recap. As I mentioned before, our approach to CRISPR is using ribonucleoproteins, also known as RNPs, and it's the most accessible form of CRISPR to date, because here you're using an RNA oligo, so no cloning or molecular biology knowledge is required. All you have to do is combine the RNA oligo with Cas9 and transfect into cells. It is really that easy. The RNPs show high editing efficiency, and allow you to perform CRISPR experiments as quickly and as efficiently as possible. Thanks. Next slide. So one of the biggest challenges when creating a knockout is that using a single guide doesn't always equal a true functional knockout. As if the indel is of a multiple of 3, then the reading frame will still be retained. So what this means in practice is that most researchers will test 3 to 5 individual guides and then move forward with the one that works best. But that means that every researcher is performing an experiment three to 5 times at a minimum, before they're able to get a viable knockout population. So this is something that our R&D team wanted to find a solution to. And what they found is that if you use 2 or more guides simultaneously in a cell, you actually get a fragment deletion rather than an indel. And a fragment deletion is more likely to correlate with a true functional knockout. And what makes this multi-guide technology unique is that we are specifically designing up to 3 guides to the same early exon, with the max distance being 150 base pairs between the first and the last guide. And that way, the guides are able to work synergistically together to create a controlled fragment deletion, and can be detected using a single Sanger sequencing reaction. And we call this technology XDel. And this is different from a lot of the other multi-guide technologies in the market, where they will design guides to random locations of the gene and hope for the best. Here, we can get our guides to work together because of the close proximity between the guides, and they're designed to work synergistically. Next slide, Travis. So in order to show you why we've taken this multi-guide approach, here is a graph where we look at indel frequency, so the ability of the Cas9 and guide to cut double-stranded DNA and create an indel, and functional knockout score, which we define by percentage of frame shifts and indels that are either greater than 20 base pairs or non-derivatives of 3. So what we've done here is we've taken our multi-guides, split them up into single guides, and transfected them into cells. And you can see when it comes to indel frequency specifically, there isn't a big difference between indel frequency in single guides and multi-guides. But where you see a much bigger change is in functional knockout score. And you can see that a huge variation between the best and the worst single guide, with the best single guide working well, but a lot of single guides just not giving good editing efficiency. While when it comes to our XDel multi-guides, you're able to see a much higher, much tighter, and much more consistent editing efficiency. And this is the technology that we now recommend using to generate knockout. And it's really good whether it's your first CRISPR experiment and you want to generate your first knockout, or if you want to save time by not testing multiple guides, or if you want to knock out multiple genes, XDel is really for you, and can be a valuable asset to your CRISPR toolbox. Next slide. So we offer this XDel technology in both tube and plate format. So our tube formats are called gene knockout kits, and they're really there if you want to generate knockouts one gene at a time. Alternatively, we have arrayed CRISPR libraries, which are there for people who want to perform more high throughput CRISPR screening. So anything from 20 genes all the way to the full human genome, so near 20,000 genes. We're able to create libraries for you. All of our libraries are customizable, so you control which targets are knocked out. So speaking of which, I'm now going to briefly discuss arrayed CRISPR screening in a bit more detail, which is crucial for people who want to perform multiple knockouts in one go. Next slide. So there are two types of CRISPR screening, pooled and arrayed. In a pooled screen, you're transfecting all of your guides into a single cellular population. You add a treatment of interest, and then use NGS to deconvolute which target has been affected at a single cell level. Arrayed screening, on the other hand, involves knocking out each gene individually in multi-gene plates. Thanks. So both pooled and arrayed screening have advantages and disadvantages. Pooled screening has an easy assay setup and is typically cheaper. However, it's only really useful for binary assay outputs, such as cell death or cell proliferation or drug sensitivity. Arrayed screening, on the other hand, has a more complex assay output, but it's much easier to interpret the results, and it's more amenable to a complex assay readout, such as differentiation, protein-to-protein interaction, or biochemical assay outputs. So which one should you use? Well, it really depends on your experiment, and it depends on your kind of project end goal and what sort of assay you want to run on your CRISPR libraries. A lot of people will perform a pooled library at a whole human genome level and then move over to an arrayed format when they've got a hit list of a few hundred or even 1,000 genes. At EditCo Bio, we focus solely on arrayed CRISPR screening. Next slide. So we can design and generate CRISPR libraries for you in 96 and 384 well plates, customizable to your work. So you pick the layout, you pick the gene list, you can pick the plate types, the control, and the number of replicates. It's fully customizable to your projects. And we can generate a library in humans within a week. So our typical turnaround time for a small library, which is up to 768 targets, is around 7 days. For a whole human genome, we can turn this around within 6 to 8 weeks. And we typically see very high editing efficiencies. So our users report anywhere between 85 and 95% editing efficiency when using our XDel knockout libraries. Next one. So this is an example of a primary dendritic screen that was done at the Research Institute using EditCo screening libraries. So you can see, and what you're looking at is consistently knocked out genetic targets using large fragment deletions. So everything on the graph on the left, everything in color represents a knockout, which each bar is the editing efficiency of each gene being knocked out in a different dendritic cell. And the editing efficiency was analyzed using NGS. On the right-hand side, you can see a volcano plot showing editing efficiency on the x-axis. And as you can see, you get high editing efficiency around 97% among all 288 targets. And what's nice about this is that this is data directly from customers using our XDel products, where they're able to get high editing efficiency. and is not data generated by us. So you can see that both, using our XDel technology, you're able to get high editing efficiency, both in our hands and and in the research community's hands. Now, a frequent question I get asked is, what if I don't have the capacity or the expertise to perform a CRISPR edit myself? And I'll now hand over to Travis to take it from here.
Awesome, awesome. Well, thanks, Niraj, for the introduction and to CRISPR and some of our products, at least from our reagent side of the portfolio. Hi, all. As Niraj said, my name is Travis Hardcastle, and I'm Director of Product Management here at EditCo. And I wanted to take some time to really walk through and introduce everyone to our cell line engineering services, so the services that we're able to offer here at EditCo. So at EditCo, we're a team of scientists, engineers, and operators that's based in Redwood City, California. And when we started EditCo, we kept hearing the same, consistent frustrations from researchers, right? Projects were getting stuck in the lab for months, or their in-house attempts to do editing, as Niraj talked about earlier, with some of the pain points, were slow, inconsistent, and non-replicable. The pain point was holding back entire sort of discovery programs at that point. So we decided to build something that was a little bit different, right? We wanted to build a purpose-built, state-of-the-art, automated cell line engineering facility to make genome engineering fast, reproducible, and scalable. We see ourselves really as that trusted partner for many biotechs, pharma, and academic groups who need these precise cell models to really drive and move their science forward. But before we get into the details of our engineered cell services, I wanted to set the stage for why they're so important. So engineered cells are used everywhere in discovery, as you probably know. They become really that backbone of modern research. For loss of function studies, knockouts reveal gene function and synthetic lethality interactions, which we will discuss a little bit more detail later on in the presentation. Oncology programs really rely on these tools to quickly identify which genes drive tumor survival. For transgenic reporters, you can really monitor pathway activity, say transcriptional factor activation, at scale, which is crucial for screening and biomarker discovery. For disease models, we can engineer substitutions or deletions that really mimic patient-derived mutations. For example, rare neurological SNVs that would otherwise take years to study can now be, modeled within a matter of weeks, as Niraj talked about earlier with some of these tools that we have at our disposal. An example of this was the work that we did for the NIH project as a part of their Indy project. I won't talk about that in detail. We're happy to come back later and talk about, give you more details as it relates to that project, but that's an example of this large-scale genome engineering project that we're able to do. For things like tags, like HiBiT, or even Halo tag, we can measure protein abundance in real time. So for example, one pharma partner that we worked with uses our HiBiT tag cells to build degrader potency curves, all in a single assay. So in short, CRISPR really unlocks engineered cells at every stage within that drug discovery pipeline. Now, I think it's important to pause here a bit because when you're working with biology, as a lot of you probably know, the level of complexity is staggering. When many of us were first taught about the cell, whether it would be in school or while we're getting our training, we were often taught the simple analogy of an egg, right? That nucleus was the yolk, the cytoplasm was that sort of white egg white cell around the outside, and the shell was that outer membrane, right? And this picture was useful as an introduction, but it's not how the cell actually functions, and we all know that now. What you see here on the screen is that eukaryotic cell panorama, right? This was really built from high resolution imaging and molecular data. Each one of these illustrations, whether it be protein sorting in the Golgi, vesicle transport, nuclear pore transport, is packed with thousands of these proteins, right? And each of these is drawn to scale. Instead of that empty space, what we see is this incredibly crowded and dynamic environment within the cell. So every protein in this image is not just floating randomly. They're interacting, binding, and moving in ways that are finely tuned. The Golgi, for example, isn't just stacks of membrane. It's that dense network of protein complexes that's orchestrating processes involved in, coat proteins, motors, and receptors. Vascular transport is not just little bubbles moving cargo. It's highly choreographed process. that works together. And nuclear pores aren't just holes in the membrane, they're massive machines made of hundreds of proteins that regulate exactly what gets in and out of the nucleus. So what this tells us is that biology operates at a level of precision that goes far beyond those textbook diagrams. The interactions are dense, highly coordinated, and incredibly sensitive. And if you want to study biology, especially in the context of drug discovery, you need tools that respect that complexity. Because if you put noisy, imprecise models in, you'll get noisy data out. But if you start with precise engineering using a cell engineering platform, where whether that's with tags, precise knock-ins, or carefully designed knockouts, you can generate models that truly reflect the intricacy of biology. That's the philosophy behind our approach here at EditCo. Precision in equals precision out. So the natural question is, how do we actually deliver quality cell models at EditCo? Not just every once in a while, but reproducible and at scale. So the answer, as Niraj alluded to earlier, is our automated editing platform. What you're seeing in this video is one of the workhorses of that platform. Every day, automation performs cell maintenance and imaging across hundreds of projects simultaneously. That includes programmable media changes, cell passaging, clone picking, and genotyping with our high quality in-house NGS workcell. And every single move is tracked, so project provenance and data integrity are maintained throughout the whole process by design. Now, why does this scale matter? It's not just about being able to run more edits in parallel, although that's a part of it. It's about reproducibility and confidence. When you run projects one at a time by hand, every scientist has a slightly different technique, a slightly different timing, and you get this variability that's introduced within the process. That's the artisanal side of science. But when you're making decisions about which drug target to pursue or when to invest in that next stage in development, variability costs time, money, and confidence to a lot of researchers. So our platform is built on removing that variability. By standardizing every step, we can deliver consistent quality across all the projects, whether that's in an immortalized cell line, or an iPSC, or even a difficult to work with primary cell model. And because the system is scalable, we keep turnaround times and even as project volumes grow. In other words, automation doesn't just make us faster, it ensures that every customer, whether they're running one project or 10, gets the same level of precision and reproducibility. That's why the scale matters. It transforms cell editing from one-off experiments into a dependable capability that you can build your drug discovery pipeline on. So now I want to take a pause here for a moment and take a step back from the process and the biology side of things and take a look at the economics of CRISPR, because that's something that many researchers often underestimate, right? So this slide comes directly from our CRISPR benchmark report. And in this report, we surveyed researchers across industry and academia on how many attempts it actually takes to produce a successful edit when they run CRISPR in-house in their own lab. And if you look at the first column, labeled one attempt on the left there, this is the best case scenario. The total time invested is about 106 hours, and the all-in cost, including labor, reagents, all the consumables, comes out roughly to around 4,300 US dollars. But this assumes everything goes right the first time, which we all know that never really happens in practice, right? The report shows that most researchers require multiple attempts in achieving a successful edit. When they move to these into maybe four attempts, that four attempt scenario, the total time jumps to about 289 hours, and the cost rises sharply to around 12,000 USD. Now, look at the center of the slide, the average researcher experience, right? This is based off the data that we were able to collect. On average, it takes about 7 attempts to get a successful CRISPR edit. At this point, you're looking at around 427 total hours invested, and then an internal cost of more than probably $18,000 at this point. And still, that's just for one knockout or knock-in, particularly knock-in these days. This is simply the cost of getting the edit to work at all. For groups to push even further, attempting 10 rounds of optimization, the cost balloons to more than 25,000 and over 650 hours of effort. That's months of people hours and tens of thousands of dollars tied up in uncertainty. The point here is that not every research group or researcher is doing anything wrong. This is simply the reality of manual CRISPR workflows at times. They are slow, they're variable, particularly in knock-ins, and expensive. And this is exactly the pain point that EditCo solves. When we say our XDel or knockout services are priced attractively and delivered reliably, it's because we're already internalized all the automation, optimization, and QC that eliminates the repeated failure cycles. What takes internal teams 7 attempts, we deliver with consistency and efficiency the first time. So the takeaway really is quite simple. Doing CRISPR in-house comes with a hidden cost that most labs dramatically underestimate. And partnering with EditCo gives customers predictable success without repeated trial and error processes. So one of the most important questions we get from researchers, especially those working in iPSCs, is about reproducibility. If you can't trust your editing results from experiment to experiment, then it becomes extremely difficult to interpret biological outcomes or to run these high throughput studies, right, that Niraj was talking about. This slide really shows two pieces of data that illustrate how consistent and reliable EditCo CRISPR editing is, even when working at large scale. On the left, you're looking at a 384 well plate, where each well represents an independent edit performed as individual technical replicates. What's notable here is the high and tight distribution is across the entire plate of edits. Almost every well falls within this high editing efficiency range, with only minimal variation between each of the replicates. This is the hallmark of an industrialized workflow, such as what we have here at EditCo. We use automation and standardized chemistry to remove that variability that you typically see within manual editing. So whether you run 5 edits, 50 edits, or 500 edits, you can expect the same level of performance across all of the edits that we do. On the right, we're showing an example of the MAS3 gene, where we look at the editing profile of the cell pools at 2 different time points, week 1 and week 4. Remember, this is not clonal data, this is actually pool data. And these are heterogeneous populations, which is normally more challenging to interpret. But what you'll notice here is that the editing signature is remarkably stable over time. The distribution of indels doesn't drift or change over time, and the overall profile remains consistent. Why does this matter? For researchers working with iPSC, stability is essential. You want to know that the cells you're working with today will behave the same way weeks later, especially if you're doing downstream differentiation, screening, or any sort of disease modeling. So the takeaway here is simple. EditCo editing is not only high efficiency, it's reproducible, stable, and reliable over time. That means that more researchers can have confidence in their experiments with more consistent biological readouts and fewer failed replicates. One of the biggest hurdles with CRISPR editing, as Niraj talked about earlier, especially when working across many different cell types, is that no 2 cell lines behave the same. What works really well in one cell line may fail completely in another. That creates huge variability and, for most labs, a lot of wasted time and resources. And it co-solves this with our fully automated platform that we saw earlier. What you see here is a system capable of performing a 200-point optimization experiment, which we do with all new cell lines that we work with. This allows us to test a wide matrix of different electroporation conditions, reagent concentrations, and delivery parameters. This platform has been applied to well over 1,400 unique cell lines at this point, spanning immune cells, epithelial cells, neurons, blood-derived lines, stem cells, and many more, as illustrated in the diagram showing the diversity of the tissues be successfully optimized. Let me walk you through the charts at the bottom of the slide, though. On the left, you have an easy to work with cell line, these EMT6, which is a breast cancer cell model. You can see that almost every transfection condition yields a high editing efficiency. For this type of line, CRISPR is pretty straightforward. It's relatively easy to work with. In the middle, you see a moderately difficult cell line, 5637 cells from bladder tissue. Here, only a subset of conditions yield strong editing efficiencies. Without optimization, most researchers would land in a suboptimal sort of parameter set and assume that the line is difficult or low efficiency. But with automation, as we have in EditCo, we quickly isolate the top performing conditions and deliver highly efficient edited cells every time. Now, the most important example is on the right there, these THP1 cells. I've worked with these cells. They're an absolute nightmare to work with. They're a blood-derived line traditionally considered one of the most challenging models to edit. If you use the standard protocol that most CROs or labs rely on, you get around the 7% editing efficiency, which is essentially unusable for any sort of downstream applications. But with EditCo's optimized platform, we've identified conditions that deliver 82% editing efficiency. That's more than a tenfold improvement. That demonstrates a key point. Success isn't about luck or trial and error. It's about systematic, automated optimization, which we do here at EditCo. Because we've optimized more than 1,400 cell lines at this point, we already understand how different tissues respond to CRISPR delivery. And when we encounter a new line, our platform readily identifies optimal conditions, ensuring our customers get high editing efficiency regardless of the complexity. This is why EditCo has become the go-to partner for many researchers working with challenging cell types or scaling large engineering projects. Our automation ensures not just success, but consistency and reproducibility across all editing campaigns or all editing projects. So before we close this section, I want to give you a quick summary of why our partners choose EditCo for their genome engineering work, especially when precision, scale, and turnaround time matter. We anchor everything we do around these four core pillars of value. First, EditCo delivers extremely consistent editing performance. Across our knockout services, we see greater than 98% average on-target editing efficiency, and we maintain over 90% of cell viability across all the passages across these cells. This combination of high efficiency editing with healthy, stable cells is one of the key reasons our engineered models perform so reliably in downstream assays. Whether teams are doing screening, mechanistic studies, or disease modeling, they get clean, reproducible data rather than a noisy data. Second is speed. Our XDel knockout pools, as Naresh talked about with our XDel technology, are typically delivered in 3 to 4 weeks. And even our XDel knockout clones, which involve the single cell cloning that Naresh talked about earlier, we can complete in anywhere from 8 to 10 weeks. For customers, that's drastically or dramatically faster than attempting edits in-house or through a traditional CRO route. And speed matters when project timelines, grant cycles, or competitive windows are tight. Third, EditCo is built for scale, as we talked about with our automation. Our platform allows us to run hundreds of edits in parallel, which is critical for projects like large variant panels, synthetic lethality studies, or genome-wide perturbations, or multi-target exploratory work. We also maintain access to well over more than 1,000 cell lines, including many hard-to-edit cell lines. That means customers aren't limited to the narrow set of easy-to-work-with lines. We can support biology that matters to them. And finally, cost efficiency. Because of our automation, expertise, and reproducibility, we act as a cost-efficient partner compared to the real, often underestimated costs of internal CRISPR workflows, which we discussed earlier. Customers avoid that repeated trial and error cycle and drive up both time and budget. Instead, they get predictable success and predictable timelines that ends up being significantly more economical in almost every scenario. So when we say EditCo is a partner for rapid, precise, reproducible, and cost-efficient genome engineering, these are the pillars we are referring to. They are what enable our customers and researchers to move faster, reduce risk, and generate data they can trust. So now I want to shift our attention specifically to our CRISPR knockout services. So Niraj presented this slide earlier, but I wanted to remind everyone how XDel actually works and why it's fundamentally different than traditional single-guide CRISPR editing. What you're looking at here, as we saw, is a comparison between the wild-type allele and the result of our XDel multi-guide knockouts. In our approach, we designed those 3 guides in parallel, as Niraj presented, all targeting distinct positions across the same exon or genomic region. When these guides are cut simultaneously, the cell's repair mechanism joins the ends incorrectly, creating that large fragment deletion that Niraj talked about, instead of those small indels. So why does this matter? Because these fragment deletions are far more reliable at generating these complete knockouts, as we saw the data for earlier. Single-guide CRISPR often relies on small indels to disrupt that reading frame, but the indel is in frame, and the cell can use alternative splicing to get a residual protein expression. That's a huge problem for any sort of functional assays. So you can see in the sequence traces that the wild type alleles are intact, while the XDel pool shows a clean, reproducible dropout of that targeted region, representing that fragment deletion event that we've talked about. This gives two advantages, right? Higher editing efficiency, because we're not relying on one single cut, and cleaner biological outcomes because that target gene is removed rather than simply disrupted. And this sets the stage for that next slide. We're going to see how this translates into persistent protein level depletion across time. So now here's the real proof of how the XDel strategy works with our engineered cells services. What you're looking at here is a Western blot data across 5 different target genes, measured at multiple points within an XDel edited U2OS cell pool. In each panel, the bands outlined in purple represent a knockout condition. You'll notice that in every case, the protein levels are drastically reduced, if not completely absent. And importantly, this depletion is stable across multiple time points that we measured. This is a critical point. Many CRISPR editing methods can show a temporary reduction in protein expression immediately after the editing, but as the cells grow in passage, incomplete edits or mixed alleles can allow for proteins to reemerge, for the protein to reemerge. This is a big challenge in pooled experiments, particularly in screens or any assays that require stable disruption of the gene. XDel avoids this by creating this large fragment deletion, as we just discussed. Once that genomic region is removed, protein simply cannot be produced, and as you can see here, that knockout signal stays clean and persistent over up to 21 days, I think is where we measured this here. This gives researchers a much greater confidence that the downstream phenotype, whether it's viability, signaling changes, metabolic shifts, or synthetic lethality effects, is generally due to the loss of target gene and not genetic drift in that panel or in that partial editing. So in summary, XDel delivers high efficiency genome disruption, maintains a long-term protein depletion, and produces a stable, reliable knockout pool suitable for both short-term and long-term assays. And with that foundation, let's move into a case study that we did with Everest, where the same XDel technology enabled the validation of dozens of synthetic lethal gene pairs, including brand new interactions in the FA and the FAC examples we'll hear about shortly. Now, I wanted to give this quick example of how XDel knockout platform is being used to accelerate discovery in one of the most complex areas of oncology, which is synthetic lethality. As many of you know, synthetic lethality has enormous therapeutic potential, but it's incredibly difficult to work with in practice. The data sets are noisy and highly heterogeneous, which makes it difficult to distinguish true synthetic lethality relationships from background noise. At the same time, discovery teams often identify large numbers of predicted gene pairs, but validating them experimentally is a major bottleneck. You need high efficiency knockouts across many different cancer lines, and the traditional CRISPR workflows are simply too slow or too inconsistent to support 20, 50, or even 100 gene pair validation campaigns. That's where the environment for one of our partners, Everest, was operating in. Their AI-driven, what they called Forbius platform, analyzed huge multi-omics data sets and screened 400 million putative synthetic lethal pairs in silico. From there, that enormous search space, they identified about 10,000 highly predictive pairs, and then they were able to narrow that down to about 50 or so targets that were both mechanistically rational and druggable, and they selected 24 priority gene pairs for wet lab validation. Their challenge was, however, how do you validate dozens of gene pairs quickly, cleanly, and with high edit efficiency across multiple cancer cell lines? This is where XDel comes into play. XDel is designed specifically for this high efficiency, multi-guide knockout perturbations at scale, instead of relying on traditional one gene at a time, one edit at a time workflow methods that often require weeks of troubleshooting and yield high variability. XDel allowed us to move directly into reliable, high efficiency knockout pool generation across a broad diversity of cell models. In this project, our XDel workflow enabled Everest to rapidly generate knockout pools in their selected gene pairs with consistent editing efficiency and speed required to keep up with their AI-driven discovery engine. This accelerated their entire validation pipeline, allowing them to test dozens of predicted synthetic lethal interactions within a single experimental campaign rather than over many months. And this is just a high-level summary. There's a much deeper biological story here, including how the work led to validation of novel synthetic lethal mechanisms, for example, with the FAK biology, or how these findings informed new medicinal chemistry programs, but we'll save that for another time. For today, the key takeaways are simple. XDel enabled scalable, rapid validation of next-generation synthetic lethal targets, supporting faster oncology target discovery and reducing that gap between computational prediction and experimental confirmation. And if this is an area of interest for you and your teams or anybody that you're working with, we'd be very happy to come back and walk through the full case study in more detail. There's a lot more beneath the surface here, including the downstream biological insights, assay results, and how this approach is really reshaping their early stage discovery oncology programs. And to finish up, I want to shift our attention into our knock-in services portfolio. So let's break down what researchers are actually asking us for. You've got SNVs or point mutations, which matter for disease modeling, things like modeling Alzheimer's variants, Parkinson's variants, oncology mutations, anything tied to patient stratification. Then you have tags in the form of luminescence tags, fluorescence tags, affinity tags. These are really used for protein trafficking, protein tracking, purification, visualization, or any sort of signaling studies. Then you have things like background tags, right? Used in PROTAC research or controlled protein degradation experiments. These are huge in pharmas doing targeted protein degradation work. Finally, you have things such as reporters or fusion genes. These range in size from 1 to 3 kb inserts, and they really allow researchers to build biosensors, pathway reporters, or transcriptional activation assays, right? The most important message here, though, is that all of these are custom engineered projects. They require design, donor template construction, transfection optimization, cell line specific know-how, and QC infrastructure that many labs just quite simply don't have. That's where we have leverage here at EditCo. We're taking the customer's most painful, slow, risky internal workflows and handing them to a company like EditCo that does this at scale every day. Now I want to highlight what is easily one of the most disruptive technologies we offer, which is our HiBiT tagging custom engineered services. So for those of you who don't know, HiBiT is an 11 amino acid peptide. So it's a super tiny peptide, so small that the protein doesn't care that it's there. It doesn't interrupt folding or localization, but when you bring in Promega's complementary protein, large bit, they reconstitute to form this bright luminescent enzyme. This gives you a real-time molecular flashlight into your protein of interest. Instead of guessing protein abundance through antibody binding or reconstructing artificial biology with overexpression plasmids, you get this fast quantitative readout directly from this endogenous locus within the genome. HiBiT basically turns any protein into a live reporter, and more importantly, without changing its behavior because the peptide is so small, it's only 11 amino acids. This is where HiBiT really comes alive, the applications it's able to serve, right? So in oncology, researchers measure protein stability, drug-induced degradation, PROTAC activity, pathway activation, things that are incredibly hard to do cleanly with classical tools. In neurobiology, you can track aggregation-prone proteins or synaptic markers with far more sensitivity than, say, a fluorescent tag. For things like virology, you can measure viral protein expression, host pathogen dynamics, and replication kinetics without any sort of antibody that often cross-react with different species. In immunology, things like cytokines, immune checkpoints, activation markers, all of these can be measured in real time with HiBiT. And for things like drug discovery, scalability is everything. A luminescence readout that can be run in 96, 384, even 1536 wells is a real game changer in drug discovery and screening. What this means is that for researchers and distributors such as yourself, the HiBiT is universally relevant. You're not pigeonholed into one niche or one department. almost every group working with proteins can benefit from a HiBiT model. I think this is a real educational moment for everyone. Most labs rely on plasmid lentiviral tag proteins, but we know that there's a problem with that, right? As Niraj alluded to earlier, with viruses or plasmids, you can have this random integration. You can have clonal variability. You can have anywhere from 20 to 100x overexpression, which causes mislocalization and inconsistent readouts. CRISPR-based tagging is precise. It edits the endogenous locus, meaning expression stays under that natural regulation of the cell. So instead of looking at noisy artificial data sets, researchers get really clean kinetics with real biology. This comparison is what unlocks pharma budgets, and they immediately understand why the current data set lacks this resolution, understand that their current data lacks this resolution. So there are many, many use cases when it comes to HiBiT that we could talk about, that we could use. But I wanted just to highlight one here, and we can come back and share many more of these. This slide highlights just how HiBiT enables lysol tracking of receptor internalization, a process that's central for many sort of drug responses or signaling pathways. So what does it enable? By inserting the HiBiT tag at this extracellular domain of a receptor, say in this case on the screen here, EGFR, researchers can use Promega's NanoGlow HiBiT extracellular detection system to measure cell surface expression in real time. Because HiBiT only produces luminescence when it's accessible to the large bit, a decrease in the signal directly reflects receptor internalization. So you don't need to use any labels or any antibodies or any sort of overexpression artifacts. So why does this matter? This gives researchers a clean, quantitative way to study receptor trafficking, recycling, and downregulation, which is crucial for understanding drug efficacy, resistance mechanisms, or even receptor desensitization. So unlike fluorescent tags or antibodies, this method is signal-specific and scalable, which makes it ideal for both high-content screening and detailed mechanistic studies. Here on the right, we're showing data from Promega's R&D team. After treating HeLa cells with EGF, you can see a clear dose-dependent loss of surface luminescence, indicating receptor internalization. This reflects real biology, native expression, endogenous trafficking, and precise drug response, without any of that overexpression noise. With CRISPR plus HiBiT, internalization becomes measurable, not just observable. That's a game changer for anyone studying receptor biology, from basic science to therapeutic development. As I said earlier, there are many, many use cases of using HiBiT that we could highlight. And myself or Niraj would be happy to come back at a later time to give more detailed presentation on just everything that we're doing with HiBiT, whether that be in some point later in 2026. But to finish up, I just want to talk about at least one specific case study outside of HiBiT, but with our knock-in services portfolio. So I want to shift to an area that's the area where high quality genome engineering is making an enormous impact, and that is rare childhood disorders. Although each individual rare disease affects a small number of patients, collectively the burden is extremely large. In the US alone, over 25 million Americans suffer from rare disease. Globally, that number reaches hundreds of millions. Here are roughly 7,000 known rare diseases, or there are roughly around 7,000 known rare diseases, and most of them are genetic. What's even more striking is that 50% of all patients with rare disorders are children. Their families, who are often spend years searching for answers, only to discover that the underlying genetic cause is poorly understood or completely unknown. One of the leading research groups working to uncover these genetic drivers is TGen, the Translational Genomic Research Institute. TGen focuses on identifying a molecular basis for rare childhood conditions such as DNM1 mutations linked to epileptic encephalopathies, mitochondrial disease, which are often present early in life and lead to progressive organ failure, or Crisponi syndrome, a severe neonatal condition involving respiratory and feeding difficulties. TGen's overarching mission is very much clear. They want to identify the genetic factors that cause these rare childhood disorders and use that information to guide earlier diagnosis, better treatment strategies, and ultimately better outcomes for the affected children. Achieving this mission requires a scalable, reliable genome engineering and iPSCs. And that's where EditCo becomes an essential partner for a company like TGen or a research institute like TGen. TGen came to us with an ambitious scientific goal: generate genome edits across a broad panel of iPSC targets to study rare childhood disease variants. Initially, they had identified 60 targets across numerous rare diseases, really representing a diverse and scientifically important set of mutations. This was not the ideal experimental design, this was their ideal experimental design, a broad panel allowing them to study many disease pathways in parallel. However, in reality, they face significant constraints. Traditional genome editing approaches, as we've talked about, are slow, costly, and difficult to scale, especially when dealing with iPS cells. As a result, TGen had to narrow their focus to about 5 targets, all within mitochondrial disease. And even that reduced scope, stretched their available bandwidth and budget. But here's where the story changes. Using EditCo's industrialized CRISPR platform, we were able to eliminate those traditional limitations. Instead of producing only 5 edits, we successfully generated all 60 edits across the intended disease pathways, restoring the full scientific scope of TGen originally envisioned. And we did this in just under 6 months, or just about 6 months. This outcome illustrates the transformative impact of scalable CRISPR engineering that we're able to have at EditCo. Without EditCo, this project would have been limited to small, incremental progress. However, with EditCo, TGen could execute the complete sort of scientific plan they wanted from the very beginning. So to understand why EditCo's involvement was so crucial, it's helpful to look at the core challenges that TGen faced. First, iPSC editing is inherently difficult. As we've talked about, it requires optimization of delivery methods, careful clonal expansion, and deep QC. Many labs simply don't have this infrastructure to run dozens of iPS editing simultaneously. Second is limited bandwidth. TGen scientists are experts in discovery biology, not in high-throughput genome editing. Their time is best spent analyzing disease mechanisms, but instead they were constrained by the manual effort required for genome editing. Third is outsourcing restrictions. Some of TGen's collaborative agreements limited how they were able and how they could outsource work, which made scaling through traditional CROs impossible. Now let's look at EditCo solved each of these challenges. Our expertise in generating high-quality iPS SNVs at scale meant that TGen could focus on discovery, not technical execution. We handled the editing, QC, sequencing, and clonal isolation, freeing their scientists to focus entirely on understanding disease biology. Because we operated within true industrial capacity, TGen was able to maximize the impact of their funding and internal staff. Instead of working within traditional bottlenecks, they gained the ability to pursue full scientific questions without compromising scope. And finally, our platform allowed them to study more targets without incurring traditional cost and timeline penalties. Automation and batch parallelization removed the assumptions that more targets equaled more costs. Instead, scale became an advantage. This is what it looks like when researchers institutes is no longer constrained by bandwidth, editing difficulties, or outsourcing limitations. EditCo became not just a vendor, but a strategic partner, enabling TGen's mission. This slide really visualizes that transformation that took place. On the right, you can see the reality that TGen brought that thought they were stuck with 5 edits, all within one disease area, shaped by feasibility and cost restrictions. It's a limiting research program, and while valuable, it doesn't reflect the full range of biological questions they help to answer. On the left, you see the outcome achieved with EditCo. All 60 targets successfully edited, spanning numerous rare diseases, all completed within 6 months. This wasn't just a technical achievement. It completely reshaped TGen's research strategy. Instead of narrowing their scope, they expanded it. Instead of incremental progress, they moved forward with comprehensive data that now allows them to investigate rare disease biology at the depth and breadth that they previously thought was unrealistic. For TGen, this success reinforced the industrialized CRISPR engineering is not simply faster, it's a fundamental change that's possible in rare disease research. At the end of the day, the reason this work matters is simple. The more variants we can study, the more patients that they can help. By partnering with EditCo, TGen dramatically accelerated their discovery pipeline. They were able to target 12 times more variants than they could on their own. They expanded their scientific investigation across multiple rare diseases rather than being limited to a single category. And even if only 25% of those variants led to actionable insights, TGen estimates that they could help 2 to 3 times more patients than before. This is the real world value of scalable CRISPR engineering: better diagnostics, better therapeutic targets, and deeper understanding of genetic mechanisms driving disease. And here you see a quote from Matt Heumann, professor of neurogenomics at TGen. He highlights that a high throughput standardized engineering platform is essential for scaling functional genomics and integrating these insights into diagnostics and therapeutic development pipelines. This partnership with TGen demonstrates that when genome engineering stops being a bottleneck, rare disease research can finally pursue its full scope of science needed to make meaningful impacts on patients and their families. And with that, I think we're done for at least what we're going to present today. And Niraj and myself will be happy to answer any questions. We'd also be happy to come back and dig into any more of these case studies or additional case studies in the future as well.